So this comes at the expense of having to truncate some of the stuff in the next lecture, but that's okay. So what I want to just go over a little bit is this idea of effective population size, because it really is very fundamental in population genetics. You shouldn't get away with the idea that it means anything except a calculating device for dealing with genetic drift. There isn't something out there which you can go and count and say that's the effective population size. It's something which enables you to write down, given a certain model, the rate of genetic drift. So in the Wright-Fisher model, the rate is equal to 1 over 2n. So that's if you have this very, very simple model of random expected contributions to the next generation with no distinction of sexes, etc., discrete generations. So under that very simple model, various different measures of the rate of genetic drift, which I'm not going to go into, all come out with the same thing. It's equal to 1 over 2n. And that includes the mean coalescence time for a pair of alleles sampled from the population. That's equal to the inverse of the rate. It's equal to 2n. So the question is, most organisms are not Wright-Fisher populations. They have, say, two separate sexes, males and females. The number of breeding males may be different from the number of breeding females. In addition, variation in reproductive success may not be purely random, so that there may be an average offspring number of 2, but the variance in offspring number may be much greater. There may also be changes in population size over the generations. So we need to deal with these kind of complexities if we want to have a general theory of neutral evolution. And it would be very tedious that each time you came to a particular situation that you had to redo all of the theory. So what NE does for you is to sidestep all of this. And of course, it's an approximation. One shouldn't be scared of approximations in biology, and it'll work pretty well if the population size is reasonably large. It just says, instead of the rate being 1 over 2n, it's 1 over 2ne. And ne is what we call the effective population size. And it was a concept which was introduced by Wright in 1931 in his very famous paper, Evolution in Mendelian Genetics. And that means, in particular, that the mean coalescent time, instead of being 2n, is 2ne. So what does all this mean, really? Well, it means that if we take a particular model of the breeding system of a species, if we're clever enough, we can work out an expression for NE. So let's take the simple example. And that's this thing. The case of two separate sexes. So I'm going to deal with NE in the context of effective population size. This seems to have given up the ghost here. So I'm going to just talk about the average time to coalescence, which by definition is twice the effective population size. So then we can go through, and if you're interested, the details of how you can do this are all outlined in the textbook or in various classic papers in population genetics. This one was worked out by Wright in his 1931 paper. He took the simple case of a population which is still discrete generations. There's still random variation in offspring number, so there's a Poisson distribution of offspring number for each individual in the population, and it's the same for males and females. But the difference here is we may have a difference in the number of breeding females and the number of breeding males. So if you imagine we census the population at the time they're actually producing offspring, we don't have equal numbers of males and females necessarily. And under those circumstances, if you do the algebra, it's not obvious, you have to do the calculation. The effective population size can be written either as this or as this. They're mathematically completely equivalent, as you can check. So it's 1 over 4f, 4 times nf, plus 1 over 4 times nm on the right-hand side. And it brings out the point that if nm is, say, very small, compared with nf, the effective, this term here will be very big compared with that term, and so when you turn it upside down, the effective size is actually much closer to nm than nf. And the intuitive reason for this, it's always useful in these contexts and in population genetics or any other mathematical application to science, to try and get a sort of intuitive sense of what's going on. There's a basic rule of biology of sexually reproducing organisms that you get half your genetic information from your mother and half your genetic information from your father. So if you think about it, even if you only have 10 breeding males in the population to 1,000 breeding females, half your genetic information, which is going to the next generation, is being channeled just through those 10 breeding males. And so that's why they have a dominant effect on the action of genetic drift, because it's a sort of bottleneck of population size, which those genes have to go through in order to get to the next generation. And in many species of animals, there's strong sexual selection, so only a fraction of the males in the population are successful as fathers. And that's why it's quite realistic to take into account the possibility that NM may be very much less than NF. There's an exercise in the problems which brings that out. Does that help you now? Is that a bit clearer what we're talking about? Similarly, we can relax the assumption that there's purely random variation in offspring number among individuals. And in that case, this was also worked out by Wright. The effective size is equal to 4n divided by the sum of the variance in offspring number plus 2. This is, again, it's an approximation. It's only valid if the population size is reasonably large. And you can see that if we have a Poisson distribution, a basic fact of mathematics is the mean of the Poisson distribution is equal to its variance. So the variance here becomes 2, so we get 4 divided by 4. It gives us NE is equal to N. So in that case, the effective population size is just the same as the number of breeding individuals. We're making no distinction here. If there's two sexes, they're equal in number and have the same variance, which simplifies life a lot. But this brings out the important point that if you have a large variance in offspring number, you can have a much smaller effective population size than the actual number of breeding individuals. So if we do the calculation of this model, it's not based on observation, this is a calculation based on a model, that's the expression for the effective population size. And finally, but perhaps most importantly, population size in reality is not likely to be constant. If we have a series of generations going back in time from the present, so this is the population size in one generation back, 2 generations back, and so on, there's a subscript n here missing. I apologize for that. So that should be, last one is the last in the series of n generations, 1 over n subscript n there, not n. What we do is, when we do the approximation to the rate of genetic drift over that time series going back in the past, we find that the rate of genetic drift is well approximated by using the reciprocal of all these numbers and taking the average over the n generations in question. And again, rather similarly to the male versus female case, if we have a very small number of individuals in any one generation, this means these go right up compared with all the other numbers, and so those are kind of dominating the process. It's the very small periods where the population size is very small that disproportionately influence the effective population size. And the most spectacular example of that is when you have a species which goes to, say, a new island or something, colonizes a new location. There's a small number of individuals to start with, and then the population expands. But that period where there's a bottleneck of population size can often have a strong influence on the variability that you see many generations later. Okay? So again, emphasize, you calculate this from the model. It's not something you can go out there and count. It's a mathematical construct which gives us a good approximation to the rate of genetic drift, in this case measured in terms of the mean coalescent time. So when one's talking about genetic drift and you look at papers on neutral theory of evolution and stuff like that, you'll see this NE coming in, and people often don't say what is the model that generates NE. You can forget about that. You can just say, I'm going to assume there's an NE. I'm going to put it in my equations. I don't care what the actual biology that's generating it is. Now, that's a bit naughty because you should really care about it, but you don't have to. And you can make this more and more complicated if you're really into masochism. You can look at sex linkage. You can look at the way maternally inherited genomes behave. You can look at sex differences in offspring number. You can do complicated things like overlapping generations, like human populations, where only a certain fraction of the population are breeding, but the time at which offspring are being produced is distributed over a large number of ages. And that's a difficult one, actually. It's not fully solved at the moment. In most cases of biological interest, the effective population size is smaller than the number of breeding individuals we see at the moment. So it's most likely, if you take the human population, if we go back in the past, population sizes were very much smaller than they are today, and so the effective size of the current human population, if we're interested in looking at genetic variability in the current human population, actually reflects mutations which arose many, many generations in the past. So we have to look backwards, and it's that harmonic mean, as it's called, over the past thousands of generations that's important in determining the current level of genetic variability, not the vast numbers of people who are alive today. Well, you could, we'll come to, I'm just going to give you a calculation of what the effective size of the human population is, mainly for entertainment purposes, but it's something which people do, and then we can discuss why it is what it is, very briefly, because otherwise I'm not going to get to my next lecture. Okay, so we can use this, you see, in the following way. This is what I had to skip. If we have an estimate of genetic diversity, it can be pi or theta w, it doesn't really matter, we can relate that to our observed diversity. So on the right-hand side, 4Ne times u is the predicted amount of pairwise diversity in the sample. And I derived that using the pairwise coalescence time model. So under the infinite sites model, the pairwise diversity per nucleotide site is equal to 4Ne times u. And that's a parameter which is often called theta, the scale mutation parameter. So for humans, okay, pi, we estimate, if we average over large numbers of independent loci in the human genome, and we look at silent variability, the average value of that diversity is pretty much 0.001. That's if we consider, say, a single population of the individuals in this room, we compare their DNA sequences, that's roughly the number we would come out with. So that's a known number. Donald Rumsfeld's famous remark about known knowns and unknown unknowns and known unknowns. This is a known known. We also have another known, and that's the mutation rate, the average mutation rate. Obviously, mutation rate varies a lot across the genome, but again, we've taken the genome-wide average. So we have an estimate from those experiments by Kong et al. On a sample of humans. Admittedly, they were from Iceland, so they might have been a bit strange, but the mutation rate per base pair per generation is 1 times 10 to the minus 8. So we know the mutation rate. And you can get mutation rates in other ways by comparing different species with different divergence times and stuff like that. But let's assume we have at least some ballpark estimate of the mutation rate. So we can then turn this equation upside down. We know this. We know this. So u is equal to theta over 4Ne. So Ne is 0.001 over 4 times 10 to the minus 8. That comes out at 25,000. So that's an estimate of the current effective population size of the humans. And as I mentioned already, population subdivision in humans doesn't have important effects on the amount of genetic diversity. So this is pretty much the effective population size of the entire species. There's some problems with this, which Mike Whitlock, I expect, will be talking about in a later lecture, but I'm going to ignore those because they don't make a whole lot of difference here. So that's our effective population size for the current human population, 25,000 individuals. I mean, that's, you know, probably what a couple of city blocks in Bangalore have got that many people, right? It's an incredibly small number compared with the current human population. Okay, so that comes to your question. So, and other people have asked me this in the break. Well, what's going on here? Why is the effective size so small compared with the current breeding system? So somebody tell me the answer. Sorry? Excellent. So the answer was the population size has increased. In fact, if we look at what's known about demography, major increase in population size, total population size has been over the last 10,000 years or so. So that's a relatively short period of evolutionary time. Remember that what we're talking about here is the effective population size, which is dealing with the current DNA sequence variation in the population. Now that has to be reflecting a lot of ancient events because the mutation rates are very low, right? 10 to the minus 8. So the time scale at which one mutation arises in the DNA is 100 million years. Something will happen at that particular base pair. So we're looking back probably into the ancient past where the human population size was extremely small, and hunter-gatherer societies even now have very low population densities compared with agricultural populations, and modern industrial countries have very large populations compared with the agricultural past. I mean, in Britain, the population size in the time of Queen Elizabeth I is estimated to have been about 5 million. It's now 55 million in England. So that's a tenfold expansion over 400 odd years. And similarly in India, there's been this enormous increase in the recent population size. Population sizes have increased enormously. Probably there have also been unpleasant events in the past, like epidemics, which have reduced the numbers of individuals to very low levels. So there's been a bottleneck, a small population size, and then a recovery. So all of that says we're looking back, not forwards, at what's determining the current level of DNA sequence variability in the population. Yeah. Yeah. Well, biological populations are quite resilient. Actually, the Spanish flu was kind of a blink. The really bad bottleneck was probably back in the 14th century when there was the Black Death, which swept certainly all over Europe and killed about a quarter of the population. And that was a really big event. Even the Second World War didn't really kill that many people. They did their best, but it's as a fraction of the population, it was not too bad. Okay, so we want to think about the meaning of NE. Basically, mean coalescent time is 2 NE, so NE is actually half the mean time it takes back to coalescence for a pair of autosomal alleles sampled from the current population. So if I take an allele from me and an allele from you, that means the average time for a pair of alleles, if we chopped up our genomes into independent bits and we took the average, would be about 2 NE generations. 2 NE generations. Okay, what's that in real time? Well, it's a little bit fuzzy what we mean by generation time in the case of humans, but roughly speaking, what we really mean by generation time in this context is the average time between the formation of the zygote that makes a mother, for example, and the time when that mother produces a new zygote. And that's roughly 25 years if you take the average over all the ages of reproducing mothers. You should also take the males into account as well. So it's basically the average age of a mother if we pull out a random zygote. So that's about 25, and it's probably been 25 for quite a long time. But let's just assume it's 25 for the sake of argument. So the time back to coalescence is twice the effective population size. Effective population size here is 25,000. So that's 625,000 years if we multiply 25 by 25,000, if I've got my arithmetic right, which I think I checked it. If not, please correct me. So the mean coalescent time for a pair of random alleles from two different individuals in this room, or actually between the maternal and paternal sequences in your genome, you don't actually need, with diploid individuals, you don't need to sample a population. You can actually just sequence a whole individual and compare their maternal and paternal genomes. This has been done several times now. So the mean coalescent time is actually 1.25 million years. What's the estimated age of origin of the species Homo sapiens from its predecessor, whatever the name of the predecessor is? Anybody know from the fossil record? Sorry? No, it's about 300,000 years. So Neanderthal man, there's big fuss recently about Neanderthal man, who's this sort of sister branch of the human lineage. It died out about, they died out about 30,000 years ago. I think the split between Homo sapiens and Homo neanderthalensis was about 600,000 years. It's not that well established. Then the split back, the predecessor was probably something like Homo erectus. So this mean coalescence time goes back to Homo erectus, the predecessor of modern humans. So a lot of our DNA variants came from Homo erectus. They didn't come from Homo sapiens. In fact, some of them, because the coalescence time has such an enormous distribution, remember its standard deviation is the same as its mean, quite a lot of our genome actually variants come from the split between chimpanzees and humans, or before that split. It's a very long time. So this is where I think, this is reasonably good science here. There's not a huge amount of assumptions. What it's telling us is that the time scale over which genetic variation is sorted out by genetic drift is enormously long. And very often, when you look at a modern species, the variation that you see in that species goes way back beyond the origin of that species. This was alluded to by yesterday, excuse me, and that's causes some problems when you're trying to sort out what's a species and what's not a species from DNA data. Yes? Yes. Well, that's a complication, but we'll talk about that in lecture 3. Okay, it's an interesting and an important problem. Probably not going to affect much of this. We know that. Short answer: we know that. You'll learn that in lecture 3. That process of effects of selection at other sites in the genome is most important in regions of the genome which have very low rates of genetic recombination. And in human populations, we see that genetic variability is fairly constant across the genome, except in these very small regions where there's almost no recombination going on. So I think that's a good question you asked there, and it's an important issue in modern population genetics, and we'll come to that. But I think the answer is this is actually pretty good. It wouldn't even affect what I'm saying, because you redefine effective population size to take into account the selection at linked sites. Okay, so I'm hoping to convince you here that they actually get some interesting stuff out of these calculations, something which is really quite informative, especially if you're interested in comparing different species. You really do have to take into account the fact that a lot of the variability within one species may actually predate the origin of that species in the sense of becoming reproductively isolated from some sister species. Certainly we and Homo neanderthalensis, whatever you think of them, they have a bad press. They're all supposed to look ugly and hairy. But genetically, they were very, very similar to us. Okay, so that's effective population size. I hope that's made it a bit more interesting than my just rushing through those few slides. Are there any other questions? So I just should say, sort of wrap up here, there's a lot of stuff coming at you. I appreciate that. I would find it very hard myself to get my head around everything that's going on in these lectures, even my own lectures. I mean, I've been giving this kind of stuff for 30 years now. But, you know, if you come to it for the first time, it's difficult. You have to sit down. You have to think about it. You can't just assume you're going to get it straight out of the lecture. We'll do a few problems tomorrow afternoon, give you some hands-on experience with some of these formulae, and that might help. But you're going to have to go away and think about it. So Sidney Brenner, the famous molecular biologist, used to say to his graduate students when they came and asked him a question, go away and think about it. It's the only way you're going to, you know, do this stuff. You can't just learn it. Any other questions? So there'll be time in the tutorials, obviously, for some feedback. If you come with specific questions, I'm sure I and Lindy will be happy to try and answer them to the best of our ability. Okay, so much for genetic drift. Now we'll have a lecture on selection, which is what was promised to you. And I'm going to have to drop some of the material because of time. But some of it's already been alluded to by Lindy and others, so that should be okay. Why do we care about selection? Well, that's pretty obvious. It's the only means known to biology by which complex adaptations of structure to function can be brought about. There is nothing else. So it's a really important key component of evolutionary biology. The only alternative is to become a creationist and assume that everything is created for a special purpose. And there are a lot of problems with that. Why on earth did God create the tapeworm, you might well ask. So selection works when there are differences in fitness between individuals, to the extent to which individuals of the given genotype or phenotype contribute to the next generation, and that can happen through differences in survival or differences in numbers of offspring. It doesn't really matter from the point of view of this lecture which of these went on. I see there's a pointer here. Good. So first proposed independently by Charles Darwin and Alfred Russel Wallace, and published in a joint paper in 1858 in the Proceedings of the Linnean Society in London. So it wasn't the book, it was actually a joint paper by Darwin and a paper by Wallace on natural selection, actually quite readable papers even now. And the written record of the meeting of the Society, the chairman concluded by saying nothing of any especial note was presented at this meeting. So there you are. So, big question is how effective is natural selection in causing evolutionary change? And there's a related one, how does it affect variability within a species? I've been talking all the time as though variability is neutral, and somebody pointed out that's not necessarily true. So I'm going to talk quite a lot about how selection affects variability now. That'll be the topic of the first bit of the lecture. I'm going to make life very, very simple and easy for us all. I'm going to focus on the same old case, a single genetic locus with two variants, and we can think of this as a single nucleotide in the genome, if we like. I want to make it really simple. And this was first done by the pioneers of population genetics, R.A. Fisher, J.B.S. Haldane, and Sewell Wright. And there's a lesser-known character called H.T.J. Norton, who actually was the first person to publish equations for the rate of change of frequencies under selection in the appendix to a book on mimicry by R.C. Punnett, published in 1915, and then had a later very formidably mathematically complicated paper on what happens with overlapping generations. So Norton should get the credit for this. Those of you who are aficionados of English literature may have read Lytton Strachey's Eminent Victorians, an extremely amusing take, history of various prominent Victorians showing what idiots they were, and it's dedicated to H.T.J. Norton, who provided some funds for it. So he has his little place in history. So, structure of the lecture. Some of this is going to have to go. I'll give the basic model of selection of the biallelic locus. I'll go very quickly over point 2, because we've heard something about this already, and rather briefly over point 4, because we've already heard some of that. So hopefully we'll catch up with ourselves. We're going to use the same old model, discrete generations. We've been through that. And we're going to, again, have diploidy. It's actually a lot easier to do haploids, so it's almost trivial and not worth bothering discussing. I will come back to haploids, though. Let's just take the difficult case of diploids, A1A1, A1A2, and A2A2. And as you heard on Saturday, the problem with dealing with diploidy and random mating is that genotypes don't reproduce themselves exactly, and that makes life more complicated. I'm going to assume, for simplicity, that we've got selection acting at the level of survival. So the only difference between these genotypes is their probabilities of survival from egg to adult. Of course, that reflects, at some level, the effects of this locus on some trait on which selection is acting. But we sidestep that problem, and we just look at the relative fitnesses of the genotypes. So we have a probability that individual A sub I, A sub J, survives from egg to adult, which is WIJ. Okay? We're indexing each genotype by its own survival probability, and initially we'll assume that's constant from generation to generation in order to make life simple. Okay, everybody with me? We could put fertility in, but I'm not going to bother. So here are the sort of equations you can come up with. In some strange way, something came in from outer space here, these little green lines. I have no idea what happened here. It's something to do with Microsoft, another crime of Microsoft. So, okay, we're assuming random mating. So if we start off with A1 and A2 with frequencies P and Q, which of course have to add up to 1, then before any selections happened, we have P squared 2PQ Q squared. We have Mr. Hardy and Mr. Weinberg tell us that that's what the frequencies of those genotypes are among the zygotes which are produced by random mating in the preceding generation. So at this point, selection is irrelevant. Now we have fitnesses. We have survival probability W11, W12, and W22, which in general will be different. So then we get frequencies after selection. So what are those frequencies? Well, pretty clear we multiply P squared by W11, 2P cubed by W12, and Q squared by W22 to get the frequencies after selection. But frequencies have to add up to 1, so we have to divide through by what's called the mean fitness of the population, W bar, which is P squared times W1, 2PQ W12, and Q squared W22. So we're just taking the average of the fitnesses of the 3 genotypes weighted by their frequencies before selection. And if we do that, we then get 3 frequencies which add up to 1. Another way of looking at this is we're measuring the frequencies conditional on survival of those individuals, and so we're dividing by the overall survival probability of a random individual. But we have to have, when we work with frequencies, we have to have things which add up to 1. And you can check for yourself that the sum of these is 1. So that's why we divide through by W bar. Now, what about, so we've got the frequencies within 1 generation. As I said, genotypes don't replicate themselves exactly with sex and random mating. So we have the problem of getting the new frequencies. And what we do is sidestep this problem by calculating the new frequency of allele A1. That's just taken by averaging over these 2 genotypes. So all of these genes in this individual are A1, right? So that's A1, A1. Forget about the red line. So This contributes p squared W11 over W bar to the new frequency of A11, but only half the genes in this individual are A1, so we'll remove the factor 2 here. Got 2pq, but only half of those alleles which come out of that individual by laws of Mendelian inheritance are A1. So the new frequency p prime of A1 is this thing. We just lose the 2 here. Similarly, for A2, we take half of this and add on the whole of that, and we get the new frequency of A2. And that's all there is to it. It's really completely mindless and trivial. What we've got is new allele frequencies, and that's all we need. We don't need the diploid genotype frequencies, because in the next generation, we've got p squared 2p q squared. We actually only have a one-dimensional problem here, because we only have 2 allele frequencies, p and q, and we know those have to add up to 1, and we can ignore the genotype frequencies for the purpose of calculation. We can just work with the allele frequencies. So if I know the allele frequency, I can predict the genotype frequency. Okay? So that's why you often read that evolution is just a matter of change in allele frequencies. Of course, the genotype frequencies matter because they're generating the individuals on whom selection acts. But what's transmitted from one generation to the next is really the allele frequency. That's all you really need to understand conceptually about this. The algebraic details are really not very important. We can simplify that. I'm just going to come down to the bottom line here. The more mysterious red lines and green lines, I have no idea where this has come from. I think it's because of my change in computer. So we can define what's called marginal fitnesses. They're really just the average fitnesses for each allele. So they're symbolized by a dot here. We're just taking p times W11 plus q times W12, because there's a probability that an individual carrying allele A1 meets another A1 individual. That's p. So you get that. There's probably q that it meets an A1. It meets an A2 allele, so we get q times W12. Similarly, W2 dot is p times W12 plus q times W22. So those are sort of the average fitnesses of each of those 2 alleles in the population. We're just weighting each allele by the probability that it hits another allele and with the fitness of the genotype that you get when that happens. And of course, the average fitness is just the average of the 2 marginal fitnesses, if you check. We can then calculate an expression for the change in frequency. I'm going to work with A2 here. That's arbitrary. We could equally well work with A1, but the 2 allele frequencies have to add up to 1. So it's just a matter of choice. And after a little bit of algebra, we get this equation. PQ times the difference between the marginal fitness of allele A2 minus the marginal fitness of allele A1 divided by the average fitness of the population. Don't worry about the intermediate steps. That's just straightforward, elementary school algebra. The important point is this is the change in allele frequency of A2. It's related to this factor PQ, the product of the 2 allele frequencies, the difference in the 2 marginal fitnesses, and the mean fitness of the population. So the 2 important, let me write that on the board. So after some manipulation of this model, we've defined the following quantities. W1 dot so that's the marginal fitness of allele A1. Marginal fitness of allele A2 is W2 dot. And the mean fitness of the population, you can check this for yourself, is just the average of the 2 marginal fitnesses weighted by their allele frequencies. Delta Q is equal to PQ, write it this, times the difference between W2 dot minus W1 dot over W bar. So that's our final expression. Has two components. There's this component which reflects the action of selection. This involves the fitnesses. And there's this component here which involves the allele frequencies. This makes some intuitive sense. What it's telling us is the rate of change of allele frequency depends on two things. The level of variability in the population, that's PQ. That's a good measure of variability. If you had random mating, the frequency of heterozygotes is 2PQ. We talked about heterozygosity, 2PQ. You can think of it as a measure of diversity. So it's very closely related to pi that I was talking about earlier, except we're now just considering an individual site. So PQ is a measure of variation, okay? What's the value of PQ if either P or Q is 0? 0? Right, so what's the maximum value of PQ? Right, so if P equals a half, Q equals a half, PQ equals a quarter. So if you graph PQ against, say, Q, it looks like this. So it's a measure of how much variability there is in the population. Selection acts on variability. If there's no variability, there's no change in allele frequency, right? And things will go fastest when PQ is at its intermediate value for a given intensity of selection. So the other thing which comes in as the strength of selection, that's given by the difference between these two allele fitnesses, but we're normalized by the average fitness of the population, okay? So that this is our measure of the amount of selection. And there's an interesting point here that it's only the relative values of the fitnesses that matter. If I multiplied all the fitnesses, say, by 100, we get a times 100 on the top and times 100 on the bottom, and it goes away. So it's not the absolute values of the survival probabilities that count here, it's the relatives, the ratios of the W's that matter. You can multiply them all by any factor you like, and you still get the same change in gene frequency. So, you know, these are two take-home messages, if you like, from the study of simple selection theory, which, you know, it's quite trivial, but it's important to understand those principles. So, in particular, we can simplify life by choosing one genotype as a standard, and we can divide all the fitnesses by the fitness of that genotype, so that it ends up with a fitness of 1. So if I divide W11 by W11, we get 1. And the other fitnesses would be W12 over W11 and W22 over W11. So they'll either have a fitness greater than 1 or less than 1. We can choose whichever genotype is the standard purely for convenience. Everybody happy? That means we've actually reduced the problem further because we've removed one parameter. We only need now 2 parameters to describe the amount of selection that's operating. And this introduces the selection coefficient, which Lindy mentioned. So let's have a look at that case. This is what we call directional selection. One of the homozygotes, for example, A1, has a higher fitness than any of the other genotypes. Okay? So there's a best genotype, and it's a homozygote, A1. Now, we can then represent the relative fitnesses of the 3 genotypes, ignoring the red lines. I suffer from migraine, and this looks more or less exactly what it's like when you get a migraine. You get these sort of jagged lines in front of your eyes, but so this is not going to induce one. So I'm going to choose A1 as the genotype. So we divide everybody's fitness by this genotype. It's the best genotype, the optimum genotype, the one with the highest fitness in this case. But it doesn't have to be, but we'll take it. This one is the standard. So it's got fitness 1. This is the fitness of A1 divided by the fitness of this is the fitness of A1, A2 divided by the fitness of A1, A1. That will be a little bit less if A1, A1 is the best genotype. Let's call it 1 minus HS. And this one might be the worst genotype. It's got 2 copies of the bad gene. It's got fitness 1 minus S. So in this case, where we're assuming that A1, A1 is the good guy, S will be positive, and it's called the selection coefficient. And that's what Lindy was talking about in her lecture. But we've also got this other genotype which we have to bother about, the heterozygote. That's got fitness 1 minus HS, and H is a sort of measure of the dominance, the extent to which the fitness of the heterozygote is influenced by the presence of A2, A2. So if H is 0, okay, what's the fitness here? 1 minus HS. 1. Okay, so if H is 0, this genotype has the same fitness as that one. So A1 is actually dominant over A2. What happens if H is 1? Right, it's 1 minus S. So in this case, A2 is dominant over A1. And A1, A2 has the same fitness as A2, A2. In general, in most cases, we might expect it to lie somewhere in between 0 or 1. So you'll meet H quite a lot in population genetics. It's called the dominance coefficient. For a two allele single locus system, that's all we need, S and H, in the case of directional selection. Then we can do a little bit more algebra. Won't bother you with the details. We get an expression for W1 minus W2. It turns out that it's always positive if S is greater than 0. So that A2, so in this case, without troubling you with the algebraic details, you can take my word for it, that W1 dot minus W2 dot is greater than 0, so that delta Q is negative, and delta P has to be equal to minus delta Q, because there's no change. The sum of P and Q is always 1, so delta P is always equal to minus delta Q. So delta P is always positive in this case, and A1 will spread through the population, providing that neither P nor Q are 0. A1 will increase in frequency at the expense of A2. Now, that's not at all surprising. That's exactly what you'd expect. It's negative, and delta P has to be equal to minus delta Q, because there's no change. The sum of P and Q is always 1. So delta P is always equal to minus delta Q. So delta P is always positive in this case, and A1 will spread through the population, providing that neither P nor Q are 0. A1 will increase in frequency at the expense of A2. Now, that's not at all surprising. That's exactly what you'd expect. I'm just going through it to show you can recapture what we think of as sort of Darwinian evolution in a very simple way in this model. So this is a trivial conclusion that under this model where one homozygote has the best fitness, that allele associated with that homozygote will eventually take over the population. But there's one thing I've left out here. We haven't asked how fast that will happen. And that's really important, and I'll come back to that quite soon. Before I do that, there's another possibility. What is it? Not you. We've assumed that one of the fittest genotype is a homozygote. Does that have to be true? Right. The heterozygote could be the best genotype. Actually, it also could be the worst genotype. So we have two cases. The heterozygote is the best, or the heterozygote is the worst. One of these is interesting, the other is not so interesting. So let's do the interesting one first. That's the case of heterozygote advantage. I'm glad somebody can read the board here. Now, Fisher in 1922 was the first person to realize that heterozygote advantage would maintain polymorphism in the population. That's the paper which is really the foundations of modern population genetics. He introduced heterozygote advantage. He introduced the branching process for the survival of a mutant gene, which Lindy will be talking about. I'll say a little bit about it in my lecture later on. He also introduced diffusion equations, which are the basis of most of stochastic population genetics. So it's three wins in one paper. And of course, nobody ever read this bloody paper for years and years and years. It was published in the Proceedings of the Royal Society of Edinburgh, of all places. This was because Fisher had encountered the enmity of Carl Pearson, who was a dominant figure in British statistics. He hated Fisher's guts because Fisher was able to derive the probability distribution of the correlation coefficient, and Carl Pearson was too stupid to do that, and he resented this. So he made Fisher's life miserable for quite a long time. Fisher then grew up and made other people's life miserable as a revenge. But I mean, absolutely brilliant paper. Heterozygote advantage. Get back from the sidetrack here. Now, in this case, A1, A2 is the best genotype. The fitness of these genotypes now are lower, so we'll call this one 1 minus S, and this 1 minus T, just make notation nice. We could use the same notation, but it gets rather nasty. I won't go through the details. We can write down marginal fitnesses for W1 and W2. They're actually very simple. But in this case, we have the possibility that because W1 and W2 depend on the allele frequencies, that W2 could equal W1, in which case delta Q will be 0, even though P and Q are not 0. Okay? So here, unlike the directional selection case, W1 can change because of the change in allele frequencies, and it can equal W2, and you get no change in allele frequency. And in fact, if you solve the equation, delta Q equals 0, I'm not going to do the algebra, you get a very simple expression for the allele frequencies. The equilibrium frequency of A1 is T over S plus T, and Q star is equal to S over S plus T. So those are the equilibrium frequencies. That's the place where delta Q is equal to 0. And here we've solved it by setting W1 dot equals W2 dot and asking what values of the P and Q give you that result. And of course, P star is equal to 1 minus Q star. And furthermore, we can show that this equilibrium is likely to be stable, because if Q is less than Q star, then it turns out delta Q is positive. If Q is greater than Q star, we can show that delta Q is negative. There's some vertical line has crept in here, which shouldn't be there. That's not 0.1. That's 0 followed by a full stop. Something weird going on with the software here. I've used this slide for many times, and it's never done something like this. Anyway, so there's an equilibrium, and furthermore, if you're on one side of the equilibrium, you move towards it. If you're on the other side of the equilibrium, you move towards it. And so it looks like it's a stable equilibrium. You can sketch this out. It's always useful to do this sort of thing. Sketch the function, as Kavita mentioned in her lecture on Saturday, and this is roughly what it looks like. I'm plotting here Q going from 0 to 1. So that's the x-axis. Delta Q on the y-axis. This is the line 0, and here's our equilibrium point. It's also 0 here and here from the argument we had before about P and Q being 0. And in between, it looks something like this. So it's positive in this domain and negative in that domain, which shows us that the population is being pushed towards the equilibrium frequency from either side. So it's a stable equilibrium. Now, those of you who are into mathematics and physics will know this is not sufficient to prove it's a truly stable equilibrium, but it turns out that in this case it is. The population will move smoothly to this equilibrium from either side, and there's no funny business about oscillations. So it's a stable equilibrium. I haven't proven that. It takes quite a bit of work to do that, but trust me, it is. So heterozygote advantage is very new mutation with heterozygote advantage that's arisen in the population. Okay? So it has frequency Q. That's the allele frequency. What's the frequency of homozygotes for A2A2? Q squared. Thank you. So we get A2A2 as Q squared. What about the heterozygotes? What's the frequency of the heterozygotes? Great, 2pq. Okay, so which is going to be bigger, Q squared or 2pq, if, say, Q is equal to 10 to the minus 4? 1 in 10,000 individuals carries that mutation. Right, 2pq is going to be much bigger. This is 10 to the minus 8. This is 2 times 10 to the minus 8 times 1 minus 10 to the minus 8, which, let's face it, that's 2 times 10 to the minus 8. We can forget about the little term inside here because it's so small. 10 to the minus 8 times 10 to the minus 8 is 10 to the minus 16. That's just a number we don't have to worry about. Sorry? Sorry. Thank you. You're ahead of me. Okay, 10 to the minus 8 is a number we don't have to worry about compared with 10 to the minus 4. So basically we have 2pq is about 2 times 10 to the minus 8, and pq as a q squared is 10 to the minus 4, sorry, and 10 to the minus 8. So in general, it's q squared 2pq, and the ratio is 2pq to q squared is roughly equal to 2p over q. Or if q is very small, it's roughly equal to 2p over q, 2 over q. So it's the ratio of those two is roughly 2 times 10 to the 4. It's a huge number. 20,000 times bigger frequency of heterozygotes than homozygotes. So it's the heterozygotes that matter, not the homozygotes. If you don't learn anything more about population genetics than this is actually a really useful take-home message that when Q is small, Q squared is essentially negligible, but this one is not so negligible. So we can really forget about the heterozygotes and only concentrate. If we can forget about the homozygotes and concentrate only on the heterozygotes. It's the heterozygotes that are carrying the mutation. The others are not seen in the population. That's what the slide says. I made a bit of a meal of that one. So the implication is that a mutation that increases the fitness of its heterozygous carriers will spread, even if it has a bad effect when homozygous. Once it gets sufficiently common, selection against the homozygous will prevent its increase, and that's where the W1 and the W2 balance out. And that's actually an important point, that natural selection has no foresight. It will pick something up. If it's good for you when it's heterozygous, even if it's actually really bad for you when it's homozygous. Anybody know an example of this in human populations? Okay, great. So I don't have to dwell on this one. Classic example is sickle cell hemoglobin. It protects you against malaria when it's heterozygous. When it's homozygous, it gives you this really nasty disorder, sickle cell anemia, sickle cell disease. The hemoglobin crystals don't line up properly, and you get these sickle cell red blood cells which clog up your blood system. It's an example of a resistance to malaria because the heterozygotes are protected against malaria. That's actually also true in the case of glucose 6-phosphate dehydrogenase, that gene I showed you earlier. In fact, a deficiency caused by an amino acid mutation in that gene causes lack of enzyme activity, and that protects you against malaria when it's heterozygous. There are several examples of malaria resistance where the heterozygote advantage appears to be operating. So sickle cell hemoglobin, the homozygotes with the sickle cell allele are, in the absence of medical care, have a reduction in fitness of about 20%. So, have a fitness of about 20%. So the selection coefficient T is about 0.8. In malarial populations, the wild-type homozygote compared with the heterozygote, it's the heterozygote that's protected, has a fitness loss of about 0.2. And so you can calculate the expected equilibrium frequency of the S allele from our formula, S over S plus T. That's about 0.2. You go through the numbers. So about 0.2 squared children who are born each generation, about 4%, will actually carry the disease. They'll be homozygotes for the sickle cell trait. So this is an example of how something which is actually really bad for you can be maintained in the population because of the heterozygote advantage. Of course, the population is still doing better. If you calculate its average fitness, which we could take as a measure of adaptation, the sense that Lindy was talking about, the average fitness, if you calculate it, is actually higher. It's about 0.84 compared with 0.8, measured relative to the heterozygote. In fact, in this simple case of a single locus with two alleles and constant fitnesses, the mean fitness of the population is always at a maximum at the equilibrium, if fitness is a constant. So here are two examples with rather weak selection and rather strong selection. This is the graph, delta Q against Q, strong selection, weak selection. Here's a graph of the mean fitness of the population. In both cases, the population has its maximum average fitness at the equilibrium point. So in some sense, this means that natural selection is good for you. It increases your mean fitness. There used to be this advertisement for the Guinness back in Britain. Guinness is good for you. But this is only true under certain specialized assumptions. Constant fitnesses, random mating, it's not universally true. So you have to treat this conclusion with some suspicion. In fact, there's this famous result of Fisher that the rate of increase in fitness of any organism at any time is equal to its genetic variance. We would call it now the additive genetic variance. That's a few rather mysterious pages of the genetical theory of natural selection, and it's couched in such a way that there's a whole industry of people arguing with each other about what Fisher really meant with this. But what it actually says, and you can prove this formally, that in a single locus two allele system, the average fitness will always increase over time. It's not always exactly equal to the additive genetic variance of fitness. That's only an approximation, in fact. Okay, so what can we apply this to data on variation in population? Problem is, most variability probably involves things which are much more weakly selected than sickle cell hemoglobin. That's probably the tip of the iceberg. It makes it very hard to detect selection on nucleotide variants. There's a calculation in the book which shows you that if you have, say, a selection coefficient of just 1%, a selective difference between two classes of individuals of 0.01, you'd have to measure something like 10,000 individuals to detect with some statistical confidence that they've got a difference in fitness. So that's a problem. We can detect the big effect ones, but we can't detect the small effect ones. There are now methods for detecting selection, which you'll probably hear about a little bit in the course, from the properties of variants in populations, and I'll be saying something about this in my lecture on Thursday, if we don't get to it in the course. But because this likely weakness of most selection acting on variants in populations, we may never really know the precise nature of the selection, what's causing it. We may know that selection is going on and it's actually having a significant effect on allele frequencies, but we won't necessarily be able to pin it down in the way we have done with things like sickle cell hemoglobin. It's a big problem connecting what's causing the selection to the outcome of selection when we're just looking at DNA sequence variability. I mentioned heterozygote disadvantage. That can happen. We can treat it mathematically in exactly the same way as heterozygote advantage, except we make S and T negative rather than positive, so everything carries through, and we get the conclusion that the equilibrium is unstable. In other words, there's an equilibrium, but if you're perturbed away from it a little bit, you go shooting off towards the nearest endpoint of q equals 0 or q equals 1. So it's probably not very important biologically, but it does happen. Variants which cause heterozygote advantage are going to be thrown out of the population immediately. Now, I'm going to skip over the next bit. It was actually dealt with in the lecture yesterday, fortunately for me. Frequency-dependent selection and variable selection are ways in which we can relax this assumption of constant fitnesses. Relative fitnesses depend on the frequencies of genotypes in the population. And that can stabilize polymorphisms. Deborah will be, I think, talking about an example of this in plants, and there are quite a few other examples. So the stuff on the slides you can have a look at, but we're going to go right over it. I'm also going to go right over this one. That was also mentioned in the lecture yesterday, fortunately for me. Selection can vary in its nature, either spatially or temporally, as the state of the environment changes. And this can allow the maintenance of polymorphisms under much wider conditions than straightforward heterozygote advantage. So that can be important. We have examples of those. For example, here our old friend, the Drosophila pseudo-obscura inversion polymorphisms. Studies of natural populations by Dobzhansky back in the 1940s showed that these fluctuated in frequency in a regular cycle repeatedly over different years. So in some years, in some time of the year, one form is favored, and another time of the year, another form is favored. You can see this one is going right down in frequency, then it's coming right up. And this goes on year after year. It must be a bit boring being a Drosophila inversion. It's been like up and down, up and down. And we don't know what's causing that. They must be associated with the genetic variants which are under strong selection, but to this day, we don't know what the hell they are. Okay. Right. Time's getting on. Let's talk about the dynamics of frequency change under selection. If natural selection is a credible explanation for adaptive evolution, then we must be able to explain the rate of change. If we had a sample, we did our calculations, and we found it took 1 billion generations for a rare new mutation with a given selective advantage to spread to fixation, then I wouldn't be standing here. I'd be off somewhere else doing something more sensible probably than population genetics. But we know that's not the case. But let's see how we know that's not the case. And it doesn't actually take forever for natural selection to change the composition of the population. This was basically work done by Norton, as I've already mentioned, but in more detail by Haldane and classic paper starting in 1924. And actually people use these results even to this day in experimental evolution, where you can track changes in frequencies of alternative variants in chemostat populations of bacteria or yeast, and you can actually measure the selective advantages by using the equations which good old Jack Haldane derived back in 1924. He was a sort of semi-enemy of Fisher, and Fisher made the famous remark, why does Jack find all this algebra necessary? Haldane's papers are full of algebraic detail, and Fisher didn't like that. This is an example where people measure the fitness effects of deletions of whole genes on fitness in yeast, and basically apply something rather similar to Haldane's equations to measure the selective disadvantages. And you can see actually removing a whole gene doesn't necessarily do you a whole lot of damage, especially if it's heterozygote. Selective disadvantages are actually quite small in many cases. I'll come to the model in a moment. So these are the estimated selective disadvantages that took out genes all over the yeast genome. So that's the distribution of the fitness effects of deleting yeast genes for every gene in the yeast genome. Okay, so most of them actually have quite small effects. This is the ones which don't actually kill the yeast, so I'm cheating slightly, but the non-lethal ones, the average effect for a typical deletion of a yeast gene is quite small. So how do we do this? We want to ask the question, given an initial frequency of an allele, how long does it take the allele frequency to change to a specified new value? if we can answer that question, we know what the speed of change under selection for a single locus is. And it's actually very easy to do this in principle. I'm going to emulate Lindy and consider a haploid population, very realistic for many microbial organisms. So we just now have only two genotypes, A1 and A2. Relative fitness is now a 1 and 1 plus S. So now A2 is the guy who's good. He's got a selective advantage of S. So S is the selective advantage of A2 over the wild type. And we have frequencies P and Q. Details of the analysis are all given in the handout. We're going to shoot right over those. I'm going to give you the answer. What we find is that the ratio of QT to Q0, that's the final frequency at time t, that's the initial frequency when we start the experiment or the evolution starts, is equal to P0 over Pt times 1 plus S to the power t. So these are the A2 frequencies at the beginning and at the end. These are the A1 frequencies at the beginning and the end. And the exact solution is just this ratio is equal to that ratio times 1 plus S to the power t. So it's a very, very simple exact solution. I'm ignoring any genetic drift here, so assuming the population size is infinitely large. We can simplify that a bit. This may not look like a simplification, but believe me, it actually helps you to think about it by taking logs on both sides. So ln, in my mathematical world, is the natural logarithm. That's the log to the base e. I'm happy to tell you that this was discovered in Edinburgh by Sir John Napier of Merchiston in about 1600. And this was actually a revolution in mathematics. The idea of a logarithm is something completely new, and it was discovered by this obscure Scottish farmer, basically. Anyway, so we take the natural logarithm. That's what is the natural logarithm for those of you who are not mathematicians? Basically, the exponent of ln x is x. That's the definition of a logarithm. And e, of course, is 1 plus a half plus 6 plus, in general, 1 over I factorial. And that's about 2 point something or other, 7, is it? I can never remember. So that's the logarithm. Definition of a logarithm is if we raise e to the power of the log, we get back the function. So if we take the logs of these two things, we find that the log of qt times p0 over the log of q0 times pt is equal to t. Now we've got the t outside that expression. That's why it's a simplification, times the log of 1 plus s. Okay? So we've got the log of those allele frequencies, the final and initial allele frequencies. Now, we can go a step further and assume that selection's weak. In most cases, that's not a bad approximation. That really requires S squared to be somewhat considerably less than S. And that'll work pretty well if S is, say, 0.01. It doesn't work so well if S is 0.1. Not too bad, but not as good. And we know from the Taylor's series, and you can check this on your hand calculator, that the natural logarithm of 1 plus s is approximately equal to s. So we can go from ln 1 plus s to s. This is going the other way from what Lindy did, is what we usually do. And we then arrive, don't have to be too bothered about all of these details here. The important point is that t now is equal to 1 over s. There's this term which depends on the selection coefficient, and there's this bit which depends on the initial frequencies. But it depends on the logarithm of the initial frequencies. So it depends on the logarithm of 1 over q0 and the logarithm of qt. And logs don't change very much. When the number changes from small to large, the nice thing about a log is it stays almost the same. So providing x is positive, you've got a graph which looks, something like this. Then, of course, it goes off the rails, but we're only considering positive x values here. So the log of x is a rather flat function of x. what I call the miracle of the logarithm. The time taken, so we get two conclusions. The time taken to change allele frequencies is inversely proportional to the selection coefficient. That makes a lot of sense. But it only increases logarithmically with the inverse of the initial frequency. So that's quite interesting. So even a very rare mutation can spread in a reasonable amount of time. So we've got 1 over q naught here. That could be a big number, 1 over q naught. Q naught could be, say, 10 to the minus 6. That's 10 to the 6. It's a million. But you take the log of a million, and you don't get very much. I'll give you a numerical example. So time taken, 1 over s, multiplied by something which is not that big. OK, so let q naught and pt be 10 to the minus 6. So the allele has gone from very, very low frequency to almost taking over the population. So it's got to a very, very high frequency, essentially fixation. So p naught and qt are both essentially 1. So our ratio reduces basically to 1 over q naught times 1 over pt. And that's 10 to the 12. That's a huge number, 10 to the 12, right? Very, very big. Much bigger than the age of the universe. But we take the log of that, and we get a miserable 27.6. So our time is 1 over the selection coefficient times 27.6. So it's of the same order as 1 over the selection coefficient. OK, let's go further. Let's make the initial frequency, q naught, be 10 to the minus 7, 1 in 10 million. That's a very low initial frequency. We get 32.2 when we do this calculation, almost no change. So for this simple model, haploid selection, the time taken is 1 over the selection coefficient by something, roughly of the order of 20 for a low initial frequency. So roughly 10 times 1 over the selection coefficient, as far as order of magnitude is concerned. We're doing biology here. We're not doing physics. We're not trying to get a spacecraft to Jupiter. We're just trying to get something, which more or less goes down the road. So rough calculations are actually quite good. So that's the take-home message here for this simple model of haploid selection. Time taken to change an allele frequency a lot is roughly 1 over the selection coefficient times something of the order of 10. So 10 over s or 20 over s. So that's not so bad, eh? S, well, let's make s equal 10 to the minus 3. We're talking about now 200,000 generations. Well, that's a long time. What am I saying? 20,000 generations. Never was any good at numbers. 20,000 generations. That's a longish time, but not too bad. If the selection coefficient was 10 times bigger, if it was 0.1, then it would be only 2,000 generations. If it was 50%, well, you can do the sums yourself. We just divide the expression by the ratio. Now we can do the same thing for more complex modes of inheritance. These are more difficult, even for weak selection, and you have to use calculus, so I'm not going to go into the details. However, there's one useful result that for semi-dominant mutation, where the heterozygotes in between the two homozygotes, exactly, so the dominance coefficient is 0.5, that's more or less the same as the haploid case, except the rate of spread is 1/2 for a given S. S here is a selective disadvantage to the homozygote. So we can use the haploid results to get the case for a semi-dominant mutation in the diploid case just by doubling the times. And here's some examples. Changing allele frequencies from a very, very low value to a low value, then from a low value to an intermediate value to a high value to virtually fixation. Haploid model is the case for selection coefficient of 10 to the minus 3 that I just showed you. It's about 7,000 generations for this bit of the process. Speeds up, 4,600 generations roughly for these two bits, and then it's back to the 7,000 generations. So obviously things are going fastest in this middle period. Notice the interval of gene frequency change here is much bigger, 0.01 to 0.5. That's a much bigger range than this to this. For no dominance, as I said, we double the times. So it's exactly the same. Now the interesting bits come when we look at completely dominant or completely recessive ones. It's nearly 100,000 generations. It's nearly 100 million generations here for a favorable recessive mutation to get from a very low frequency to a low frequency. Then it speeds up, and then it goes really fast. And it's the opposite for a favorable dominant mutation, because that's just the reverse situation. So we have an interesting situation here. This is a random mating population. Favorable recessive. So we have fitness is 1 plus S for the 3 genotypes. Why is it that the favorable recessive in a random mating population is so very, very slow at getting off the ground? Sorry? So yeah, it's not present. It's only present in the heterozygous. We've done it already, actually. You've got the right answer. It's our old friend Q squared is the frequency of these guys. 2pq is the frequency of these guys. If q is small, okay, we've done this already. Q equals 10 to the minus 4. Q squared equals 10 to the minus 8. Being present in the heterozygote doesn't do any good at all, because the heterozygote has the same fitness as the homozygotes. And so natural selection can't see the favorable phenotype because it's not being produced in the population. So this is an exception to what I've been telling you. The miracle of the logarithm doesn't work here. The selection equation is more complicated. And things are very, very slow because these types, the A2, A2 types, are really not being produced very much until they get to an intermediate frequency. So in a very large population, natural selection doesn't see favorable recessives. And this has some biologically important implications. First of all, if we ignore the case of recessive favorable mutations, things go nicely. You can get mutations up from a low frequency. But favorable autosomal recessive mutations, ones which are not carried on the sex chromosomes, take a long time to increase in frequency in randomly mated populations. We've just been over the explanation for that. Now, it's interesting, biologically this, and Haldane, who was a pretty smart guy, already pointed this out in his 1924 paper, that if you look at cases where we know the genetics of mutations that have recently spread to high frequencies as a result of selection in sort of insect populations particularly, they're nearly always dominant mutations, or at least have some noticeable phenotypic effect on the heterozygote. They're not recessive mutations. But what happens if you go to the lab and you collect a bunch of Drosophila mutations that affect things like eye color or wing shape or something like that? Does anybody know the answer? You take mutations that affect phenotype, they're nearly all recessive. Dominant mutations are the exception, and in fact, the ones which you do get usually kill you when you're homozygous. So we have dominant mutations which affect bristle number or wing shape, and they're usually lethal when homozygous. The same is true for mouse, in mouse genetics as well. It's not so easy to do in humans because there's ascertainment biases in picking up mutations. So most mutations that affect the phenotype are recessive. Yet when we look at the outcome of evolution on things like insecticide resistance or pesticide resistance in plants, or this thing called industrial melanism, which I'll talk about in a moment, the mutations are dominant or semi-dominant. So why is that? Almost certainly it's because natural selection doesn't see the recessive ones. It'll pick out the odd ones which are dominant, not recessive, or at least have some effect on the phenotype. They didn't have to be completely dominant. So it's actually an example where selection is controlling the outcome of evolution, even though it's going against the direction in which variants are thrown up by mutation. Mutation is throwing up primarily recessive mutations. Selection is doing what's called, somebody called John Turner called Haldane's sieve. It's sieving out the ones which are dominant or semi-dominant and allowing data fixation. So here's industrial melanism, just to wrap up this lecture. We have two alternative forms controlled by a single Mendelian gene, simplifying a little bit. It's not quite as simple as that. But the dark form is called melanic because it's got melanin pigment in its wings. This is the normal form. And here we have a tree trunk, or at least it's a fake tree trunk, but in reality that's what a tree trunk would look like back in the good old days of merry England, when there was no industry, the air was clean, and the peasants were worked to death by the aristocracy. We won't mention that detail. But then came the Industrial Revolution. The trees got, air became acidic, all the lichens which cover the trees died out, and the trunks became black. And what you can see is that a melanic form will stand out against the light background, and the light moth will stand out against the black one. And what people observed, and England in the 19th century is full of people who wasted their time collecting insects while the peasants were slaving away in the fields. And so the museum collection, so you can actually see the relative frequency of this case increased to a very high level. The melanic form increased to a very high level over about 50 generations, about 50 years. One generation a year. So this was an example of a very rapid spread of a mutation. And Haldane was able to calculate the selection coefficient from his equations. And it's a dominant mutation. There are a few exceptions to this. If we think about X-linked mutations, recessive favorable mutations will occur on the X chromosome in males. What happens to those? There's an X and a Y. What's peculiar about the Y chromosome? Those who've done biology. Pardon? There's no genes on the Y chromosome for the most part. Deborah will be talking about that later. Why there are no genes on the Y chromosome is an interesting question. But so if you have a mutation, let's say something like the locus for color blindness, a male who's got an X chromosome and carries that mutation will have color blindness, whereas if the mutation is rare, most females will be heterozygous for that mutation and won't have color blindness. So color blindness is actually commoner in males and females. There's probably somebody, male in this room, who's colorblind, but they probably won't admit it. I had a postdoc who worked with me for some time in Drosophila until I discovered he was colorblind. It was messing things up. Anyway, so mutation which occurs on the X chromosome in males will immediately be expressed and can be seen by selection. So things go better on the X chromosome than they do on the autosomes. And Haldane noticed that himself. And there's now a whole literature about the supposed faster X evolution because of selection on favorable recessives exposed in males if they occur on the X chromosome, but not the autosomes. Similarly, if a population is not random mating, but it's doing a lot of inbreeding, like a self-fertilizing plant, you don't care about whether it's recessive or not. I'll just say one more thing, and then I'll stop. It's going to introduce what Lindy's going to be talking about. She's already mentioned it. We've made one important assumption here, that the population size is enormously large and everything just proceeds smoothly, gene frequency change under selection. But that's not actually true. And Fisher realized that early on. Even if the population size is enormously large, a new mutation is going to be present in one or a few individuals, right? So, okay, we've got 5 billion people in the world. A new favorable mutation arises somewhere. That's a lonely little mutation sitting on its own. So there'll just be a few individuals carrying that mutation. So there's a good chance that all of those carriers of the mutation will fail to leave any offspring. They could all be run over by a bus one generation after the mutation has arisen. So there's a possibility that in any one generation, carriers of the mutation will by chance fail to leave any offspring. Okay, the average offspring number may be 2. On average, everybody has 2 offspring, but some individuals have 10 and some individuals have none. So if the mutation happens to be carried in an individual who by chance has no offspring, it's gone from the population. But once it's survived this period of vulnerability to random loss, because there are a very small number of copies of the mutation, it can then, say, got 100 copies of the mutation in the population. If the population size is large, we can just work with what we've done already. So this is the problem of the chance extinction of a new mutation. I'm not going to dwell on this because we'll be hearing more about this in the lecture, another lecture. But it's important, therefore, to calculate the ultimate survival probability of a mutation, the chance that it gets established in the population. And the answer is very interesting. In the haploid case, which I've just been talking about, the survival probability is twice the selective advantage, providing we assume that variation in offspring number is completely random. In other words, we're dealing with a Wright-Fisher kind of situation. And you get the same result in a diploid randomly mating population. We replace S with SH, the selective advantage to the heterozygotes. In the case of directional selection, we often call this the fixation probability, because the mutation will go, if it survives this random loss, it will spread all the way through the population and replace the alternative. That's what we call fixation. With balancing selection, of course, we're only really concerned with it getting itself into the population, sitting there. So there's an important take-home message here that actually there's actually quite a low probability that a favorable mutation will establish itself in a population. It's about the order of twice the selective advantage for the favorable mutation in the case of a haploid, or twice the selective advantage for the heterozygous carriers of the mutation. It's a famous result in population genetics. It has one important implication. I expect Lindy will be mentioning this. It's quite likely that even if you have the same type of selection, populations will diverge genetically. So, for example, if you're selecting humans for malaria resistance, almost certainly malarial infections are something which are relatively recent and associated with the spread of agriculture. So in different parts of the world, people acquired agriculture, population density shot up, then malaria was more frequently transmitted, serious health problem, and then you evolve various types of resistance to malaria. I've given you heterozygote advantage, but there are literally hundreds of different mutations, different parts of the genome, which adapt you to malaria, and these occur in different populations. Even sickle cell mutation, we know that it's arisen at least 5 times independently in different parts of the world. It's glucose 6-phosphate dehydrogenase. There's things called the thalassemias, numerous different thalassemias, which are deficiencies of production of the hemoglobin molecules due to deletions or frameshift mutations. And it's just a matter of chance which particular mutation of which locus arose in a population. So if you have something like malaria resistance, where there are numerous different biochemical ways you can adapt to and get yourself resistant to malaria, different populations are going to have quite different genetic variants. But you're probably asking, what happens if a favorable autosomal mutation is completely recessive? Branching process, that's the technical name for what calculation I just showed you, assumes that the population size is infinite. But that breaks down because there's no fitness advantage to the heterozygote in the case of a favorable recessive. But if we ever take into account finite population size, if the population is Wright-Fisher population of size n, survival probability of a recessive mutation is now, after somewhat difficult calculation, actually first done by Haldane in 1927, but cleaned up considerably by Kimura, is about 0.8 times the square root of the ratio of the selection coefficient to the population size. And that's telling you that if the population size is very big, that's almost 0. If the population size is small, genetic drift is going to be throwing the allele frequencies around a bit, and by chance, an allele will drift to a high frequency, and so then selection will start to see it. So in a small population, like in animal breeding, for example, n is maybe a few hundred, then there's a good chance the mutation will drift through the population, get to a high frequency, and then it can get fixed by selection. So that's why you get this inverse dependence on the population size here. I don't quite know why you get a square root, but that's, some mystery of mathematics. There has to be a reason for that. maybe. I'm going to stop here. There's a lot more slides about the effect of genetic drift and selection, but I think that's going to be covered quite well in Lindy's talks, so we're not missing anything. Right, tea break. Yeah. Yeah, right. They probably will do, yes. I wish you hadn't asked that question. People tend just to try and ignore polyploids because they're really quite hard to deal with because they're very difficult genetics. Unless they're an allopolyploid, I mean, you have a cross between two species, in which case, essentially, you have two distinct genomes, and you can treat those as independent of each other. But yeah, it's when you have an autopolyploid, it's quite difficult to do the population genetics. I'm sure Deborah can tell you about it because she works on plants. Obviously, there are economic reasons for working on polyploids. There is population genetic stuff theory done on polyploids, but we certainly don't want to get into it on this course. Life is, I think you probably appreciated by now that life is difficult enough as it is without making, it even more nightmarish. So on that, I will end and go and get a well-deserved tea break.